You know the difference between scraping a B and actually locking in that A star in your biology 9700 exam? It doesn't just come down to how many flashcards you've crammed into your head. No, it usually boils down to one very specific thing: how fluently you understand topic 3.2, competitive and non-competitive reversible inhibitors. If you've been putting in the hours, but your grades just kind of aren't shifting, well, you're absolutely in the right place. In this explainer, we're going to expose exactly what those Cambridge examiners are hunting for and give you the literal blueprint to bridge that grade gap. Take a look at this cold, hard truth straight from the Cambridge Principal Examiner reports. They are practically handing us the cheat code, telling us exactly where candidates bleed marks before they even realize what hit them. Quote, "Students confuse cause and effect, and students lose marks by not showing working." It's a pretty stark warning, right? You could literally know the biological theory inside and out, but if you don't explicitly show you're working, a tiny calculation error totally zeroes out your score. But like the examiners explicitly point out, method marks can totally save you. Okay, here's our game plan for today. We're covering 1. The ultimate exam mistake. 2. Inhibitors, core principles. 3. The method mark strategy. 4. Flawed versus perfect answer. 5. An interactive examiner challenge. And 6. The essential next step. All right, section 1, the ultimate exam mistake. Let's really break down exactly where students are losing out on those top bands during high-pressure data analysis. Check out the massive red chunk on this chart here. A full 40% of the examiner warnings are all about confusing cause and effect. That happens when you mix up, say, whether the inhibitor caused a shape change or if a drop in concentration caused the inhibition. Another 35% you out there are forgetting your vital units and precise terminology. And the remaining 25%? Well, they're just relying on memorized, generic textbook answers that don't fit the actual specific context of the exam question. Basically, consider this chart your ultimate map of what not to do. Moving right along to section 2, inhibitors, core principles. So, when we're comparing competitive versus non-competitive inhibitors, you gotta remember, the syllabus is absolutely unforgiving. It demands exact definitions and a crystal clear grasp of the causal relationships. Competitive inhibitors? They literally compete for the active sites simply because they look structurally similar to the substrate. Non-competitive inhibitors, on the other hand, bind somewhere else entirely, the allosteric site, which ends up forcing a conformational change in the enzyme. Top-performing students really show a connected understanding here. They don't just list facts, they explicitly explain how that structural change actually causes the drop in the reaction rate. So let me ask you directly, why do so many students find multi-step biological pathways so incredibly hard to tackle when they're sitting in an exam? Think about it for a second. It all comes down to cognitive overload. The assessment objectives aren't just checking if you memorize Chapter 3. They're actively forcing you to integrate all these concepts with totally different syllabus areas, handling massive chunks of experimental data, all while evaluating completely unfamiliar scenarios. It's a lot. Because there's just so much cognitive load, you absolutely need shortcuts to anchor your terminology. Try this quick hack. Use the first letters of key terms to build your own custom mnemonic. Like for competitive inhibitors, you could use concentration can cure because cranking up the substrate concentration actually overcomes competitive inhibition. Create your own. It sounds super simple, but trust me, in the heat of a 90-minute exam, having a mental hook stops you from blanking on the exact, precise terminology the examiners are desperately looking for. Let's jump to section 3, the method mark strategy. Listen up, because this 5-step framework is arguably the absolute most important strategy you're going to learn today. When you're staring down a complex inhibitor question, you need to stick to this sequence. Step 1, deconstruct the question so you know exactly what they want. Step 2, extract the relevant data from all those messy stimulus materials like the graphs and tables. Step 3, select your analytical framework. Step 4, execute with clear notation. This is absolutely crucial. Explicitly write out your working. And step 5, present it with the correct terminology and units. By strictly laying out your extraction and execution right there on the page, you are mathematically guaranteeing yourself method marks. Even if you completely fumble the final number on your calculator, you are safe. Now, keep that 5-step framework locked in your mind. In just a second, I'm going to show you the exact examiner clue that flips an answer completely on its head. A clue that most students honestly just skim right past. But to make that clue totally obvious, you absolutely had to internalize the framework we just talked about. All right, section 4, flawed versus perfect answer. Take a look at the flawed generic student answer over on the left. The inhibitor slows the reaction down. I mean, sure, it's true, but it's just completely superficial. Now contrast that with the top band A star answer on the right. This student notes the specific inhibitor concentration directly from the data, uses strong comparative connectives like whereas, and explicitly states that the, quote, non-competitive inhibitor altered the active site conformation, evidenced by the Vmax decreasing while the Km remained constant. Do you see the difference there? The perfect answer seamlessly combines conceptual understanding with direct, extracted evidence from the exact context they were given. Now for Section 5, Interactive Examiner Challenge. Okay, picture this scenario. A student is looking at a graph showing a reaction that's plateauing, and they immediately write down, the enzyme has been denatured by the inhibitor. I want you to literally pause this explainer right now and predict what is the fundamental error in their data interpretation. Where's their missing analytical framework? Take a quick second. To figure that out, let's map the inhibitor concentration against the reaction rate using this experimental data table. Our hypothetical student rushed to a superficial conclusion without actually extracting the stats. If they had just checked the data, they'd clearly see this is an investigation of a reversible inhibitor. Denaturation is a permanent structural breakdown, usually because of extreme heat or pH, not the standard everyday action of a reversible competitive or non-competitive inhibitor working at optimal conditions. They totally confuse the cause of the plateau. Remember, analytical depth will always beat superficial guessing. And honestly, this brilliantly highlights the higher-order critical analysis you need to bring to the table. The examiner's correction framework lays out exactly what you must do. Identify the data's limitations, check the assumptions you're making, like assuming a plateau automatically means denaturation, provide alternative interpretations, and then evaluate how all of that affects your final conclusion. This is the deconstruct, solve, evaluate method in full action. It's exactly how you prove you're actually thinking like a real scientist and not just regurgitating a textbook page. Which brings us to Section 6, Essential Next Step. All right, here are your 3 rapid-fire takeaways to absolutely safeguard your grade boundary. Number 1, master those causal differences between competitive and non-competitive inhibitors. You really need to know your active sites from your allosteric sites. Number 2, always show you're working using that 5-step procedural framework we discussed, so you lock in those method marks. And number 3, connect your core concepts to the unfamiliar experimental context they throw at you in the exam. Always back up your claims with data extracted directly from the tables and graphs. But this brings us to a somewhat terrifying question. What if you do all of this? What if you know the biology of reversible inhibitors flawlessly, you show your math, you use all the right terms, and you still end up losing the marks? No way, right? Well, the hidden cause is actually misunderstanding the specific command words. If the question specifically asks you to evaluate and you just describe, your perfect biological knowledge is honestly entirely useless. You are going to hit a hard ceiling on your marks. Knowing the science is literally only half the battle. Knowing the specific command words that dictate the mark scheme is the mandatory survival step you've got to master next. And that is exactly what we are going to tackle in our next explainer, where we completely decode the exact command words that decide your mark band. You really cannot afford to miss it. Subscribe to our YouTube channel, Ace the Exams, at acetheexams_IGCSE to excel in your Cambridge examinations. Click on the next explainer right now and complete your A-star strategy. See you there.